Hello everyone, welcome to Muscles and Muscle Tissue Challenging Concepts Part 1, Chapter 9 of the Marieb textbook. So let's take a quick look at the different types of tissue. So it's cardiac, skeletal and smooth. Cardiac is only found in the heart. It is striated. It mostly has one nucleus per cell, sometimes two. It has intercalated discs and it's involuntary, so it has autorhythmicity as well, so that's why the pacemaker cells it can pace itself. Smooth muscle, it lines hollow organs as the main location. No striations. This one only has one nucleus per cell. It is also involuntary and it's autorhythmic. While the skeletal muscle is striated, it's multinucleated, anywhere between tens to even a couple of hundred nuclei per cell. It is under voluntary control. So in terms of the skeletal muscle, we have some connective tissue that surround it. We have three layers of it in terms of the muscle tissue, muscle as an organ. Epimysium will cover the whole muscle as an organ, and this will then connect either directly to the bone. It goes from epimysium to periosteum, or indirectly as either a tendon, so a collection of dense regular connective tissue as a tendon, rod-like, or upper neurosis sheath-like for the flat muscles. Perimysium is one that surrounds what's called a fascicle. A fascicle is a small group of muscle cells. Muscle cells are also called muscle fibers. So individual muscle fibers are surrounded by connective tissue called endomysium. So this would be an individual muscle cell, so endomysium surrounding it, a few of those muscle cells, a fascicle, and it's going to be perimysium surrounding it, and then epimysium surrounds the whole muscle as an organ. It's similar in the nervous system too. We'll talk about epineurium, perineurium, and endoneurium when it comes to the chapter 11, the nervous tissue as well. So each muscle has at least two attachment points. One is considered an origin, the other one is the insertion. The origin is wherever that connection, when that muscle contracts, it does not move or less movable, whereas the insertion is what moves. So basically the insertion moves towards the origin. So let's look at the skeletal muscle cell or muscle fiber. This would be a small sliver of a very, very long muscle cell. Some muscle cells can be even half a meter in length, depending on the person. It can be that long, right? So it runs the entire length of the muscle. So there are many muscle cells running parallel in that muscle as an organ. So first is sarcolemma. So sarcolemma is the plasma membrane of the muscle cell. Sarco means skeletal muscle, and then lemma means layer. Okay, and we will find the nuclei. The nuclei will be just underneath the muscle cell membrane, the sarcolemma. Then the next one we're going to talk about is what else is found. So the equivalent of the cytoplasm of the muscle cell is called sarcoplasm. That's a liquid component that surrounds organelle. It contains myoglobin. You can think of myoglobin very similar to hemoglobin. It is a protein molecule that attaches and stores oxygen. Glycosomes, those are your glycogen and glucose components. We have a lot of mitochondria because we do need ATP for muscle activity. The next one is a new organelle only found in muscles. It's called myofibril, right? So similar to mitochondria is an organelle that produces ATP during aerobic respiration. Myofibril myofibril is an organelle that's responsible for the contraction of these cells. So we're going to look at the myofibril in detail, or more in detail. So here in this picture, the grayish light blue would be the sarcolemma, and each one of these would be a myofibril. So we got 5 here and a few mitochondria spread throughout. They contain what are called sarcomeres. We can consider the sarcomeres, which we'll talk about shortly. Sarcomeres are the structural units of skeletal muscle. Surrounding the myofibrils, we have specialized endoplasmic reticulum called the sarcoplasmic reticulum. The main function, or the only function really, is to store calcium. So it stores the calcium in this like a little pocket, and then it releases it into the sarcoplasm when the stimulus to contract is reached, which we'll talk about as well. The sarcoplasmic reticulum will dilate out. Okay, so the sarcoplasmic reticulum in this picture would be the blue, and at the ends it will dilate out terminal cisternae, right? So you'll have 2 of those terminal cisterns, and then we have T-tubules or transverse tubules. These are basically transverse invaginations of the sarcolemma. So they're like little tunnels that go through the cell. It contains extracellular fluid. So when we have an action potential, a nerve impulse signal that passes down the membrane, it can also go into these T-tubules and come very close to the myofibrils themselves. Okay. So sarcomere itself is the area between 2 Z discs, and I will talk a little bit more about them shortly as well. It is a functional unit of skeletal muscles. In terms of what they are made up of, so myofibril is the organelle. Myofilaments are even smaller. Those are the filaments that make them up, right? So there are 3 types of filaments: thick, thin, and elastic filaments. So our skeletal muscle and cardiac muscle are striated. The striations are because of these filaments' presence. Okay, we'll talk about that in a second. So dark bands are also called A bands. The light bands are called I bands. So the dark bands basically contain the thick filaments. They run the entire length of the thick filaments, while the I band basically contains no thick filaments, just thin filaments. Okay, we'll talk about them again a little bit more detail. In the middle of a sarcomere, we will have the M line, and we'll also have what's called the H zone. H zone is part of the A band. In the A band, I mentioned that it is the entire length of the thick filaments, but we also have overlapping thin filaments. Okay, but in a certain area right in the middle, during relaxed phase, we do not have some area of thick filaments which do not have overlap. So that will be called H zone. Okay, an I band actually runs to 2 sarcomeres, which we'll describe. So in this picture, there's the zigzag line is the Z disc, and the space between 2 adjacent Z discs is a sarcomere. Okay, so let's look at some structures. In this picture, blue will be for the thin filaments, the red will be thick filaments. Thin filaments are mainly made up of actin, but we also have troponin and tropomyosin, while thick filaments are made up of myosin. Okay, and that's going to be in the middle of the sarcomere. Here is an enlarged view. So basically we have just removed the terminal the sarcoplasmic reticulum and the T-tubules, and we're just going to look at that portion. So again, the zigzag lines, that's going to be the Z discs. This is thick filaments in darker red, light ones are thin. You can see there's some overlap. Okay, and then this will be the next sarcomere, and then you can see the thick filament starting. So the I band is basically running from one sarcomere, half in one, half in the next. Contains only thin filaments, no thick filaments, while A band will be the thick filaments. Okay, here is more realistic, if you may, of what it looks like. The thick filaments have heads. We'll talk about the myosin heads. Thin filaments looks like little beads. The springy type looking structure is the elastic filament. We talked about Z discs. The A band runs the entire length of thick filaments. I band, thin filaments only, no thick filaments. Then within the A band, in a relaxed state, there is some area of A band where there's only thick filaments, no overlapping thin filaments, which will be called H zone. Okay, during contracted state, the overlap increases. A band does not change size, but the I band decreases. H zone actually disappears, which we'll talk about. M line is right in the middle, so M for middle of the sarcomere. So looking at the structures of the thick and thin filaments, each molecule, you can say, is made up of a tail, a hinge, neck area, and two heads. Okay, and it's composed of myosin. The myosin head is what's going to interact with the actin. Okay, and that interaction is a bond that's called the cross bridge. Okay, these bonds form, they detach, they reform with the next actin, and so on. Okay, so the myosin heads are binding sites for the actin. So myosin head binds to actin. That myosin head also contains a site where ATP can bind to it, as well as is broken down. So ATPase enzyme, myosin head behaves as an ATPase enzyme as well, or it has a component of ATPase enzyme. The thin filament is basically actin, which has the in the big picture we are considered mainly concentrating on the globular actin, which has a little indentation, which is the active site to which myosin heads will bind. They also had tropomyosin. They also have tropomyosin and troponin. These are considered regulatory. Tropomyosin you can think of blocking myosin, okay, while troponin will control tropomyosin. Okay, and then elastic filaments are made up of the protein titin. They're basically what they look like, the springs. So they help they help with recoil after stretch, or when they also will compress. So here in this picture will be the thin filaments. Here is thick. Thin filaments, the orange will be the tropomyosin, the little beaded, the blue beads with the indentations which are covered by the orange are the actin, and then yellow here would be troponin. Okay, and we'll talk about excitation, contraction, coupling. Calcium will bind to troponin, change shape of troponin, which causes the tropomyosin to move away, exposing the active site, and then the myosin head can bind to it. So you can imagine this is here, so the myosin head will bind to the exposed active site of actin. We mentioned about sarcoplasmic reticulum. It surrounds each myofibril. It stores calcium, and it will release the calcium into the sarcoplasm. Okay, so not into the external of the cell, extracellular fluid. This calcium does not come from the outside. It's stored in the sarcoplasmic reticulum and released into the sarcoplasm. Transverse tibials I mentioned is invaginations of the sarcolemma, and this is where the stimulation will come in. So impulse will come through here on either side of the T tubule. So the beige, or the gray color rather, is the T tubules. On either side will be terminal cisternae, so they're also called the triad. So electrical impulse comes here. They're going to trigger calcium released from the sarcoplasm. So during contraction, we call it the sliding model, because the thin filaments slide over the thick. Okay, so the overlap increases when that happens. During this time, the myosin head forms cross bridges, and then it's broken and re-established to the next subsequent actin during the phase of contraction, which results in the shortening of the sarcomere, which results in the shortening of myofibrils, which in turn results in the shortening of the whole muscle cell, which is a muscle fiber. So Z discs, the ends of sarcomere, are pulled towards the middle, the M line. I band, which has no thick filaments, just thin filaments, will shorten because we're going to have more overlap. That means there's going to be less unoverlapped, if you may, actin filaments. Okay, so Z discs get closer together because sarcomeres are shortening. H zone disappears. A bands actually move. The length does not change, but they move closer. So here's a nice picture. This would be the A band here. Okay, here's a Z disc. So this is one sarcomere. We can't even see in this picture. We're just about to see the A band next. So this will be I band. This will be the next I band. Okay, so it goes to the next sarcomere. And here is H zone, a little bit lighter than the others because there's no overlapping of thick filaments. And in this picture, it's more contracted. Here you can see there's no H zone. The Z discs, this one moved here, this one has moved here, it's moved closer. The I band is much smaller. Here it's virtually half of the I band, here the other half's here. This is almost the same as that length. Okay, so that's unoverlapped thin filaments. So when we talk about contraction, we talk in terms that they have three major phases. Activation, where the nervous system basically the action potential that comes down, the neuron will result in an action potential in the skeletal muscle sarcolemma. Excitation, contraction, coupling, this action potential that goes down the sarcolemma starts off the contraction process. And the cross bridge cycling, that's kind of the sliding filament where we are forming cross bridges, we're breaking cross bridges, reforming to the next actin, and so on. So just before we talk about the physiology, we also have to cover the anatomy. So here we can imagine this is the motor neuron coming and meeting this muscle cell. Okay, so action potential is shown in here in magenta color. It's coming down. It'll come to the axon terminal at this point because it meets the muscle. It's called the neuromuscular junction. This junction, there's a small space, so electricity cannot jump from here to the next. So what it does, it's going to cause a chemical to be released. So it's called a neurotransmitter. That neurotransmitter will go across and stimulate the other side. So here are the structures. Here is that enlarged view of this little bulb here. Okay, so this is the synaptic bulb, the end axon terminal. These are those same magenta colored action potential coming down. So synaptic bulb, they contain acetylcholine, ACH, acetylcholine are found within vesicles. Okay, and then there's a space between this axon terminal and the muscle part, which is called the motor end plate. And this motor end plate has folds, so they're called junctional folds, and that's where we will find receptors to this acetylcholine. So let's go through the activities. Nerve impulse comes in, calcium from the outside. We have extracellular fluid has calcium, low amount, but it's higher than the inside of the cell. Inside of the cell is virtually zero, so it will move in when this voltage gated channels open. Calcium moves in. The influx of calcium causes the exocytosis of acetylcholine. Okay, so acetylcholine will be released. That's going to diffuse across the synaptic cleft and bind to the receptors on the other side. Okay, this is the muscle side at the bottom. When that happens, it's going to open this channel. This channel is a cationic channel. It allows sodium and potassium to move through. If you recall from 130 or previous conversation in chemistry or in other biocases, we have a lot more sodium outside the cell, a lot more potassium inside the cell. So sodium actually moves into the cell, a few potassium do leak out, but a lot more sodium moves in. So the inside, which is normally at resting membrane potential, it's negative, it becomes less negative, let's put it that way. And we'll talk more about action potential in chapter 11. Okay, so it becomes less negative. That triggers an action potential in the muscle fiber. So here the chemicals came in, they cause sodium to come into the cell, so cell is negative at resting. When they gain sodium, they become less negative, and that will trigger off when threshold is reached. That will trigger off an action potential. Action potential travels along these is going to be the sarcolemma, the membrane of the cell. It also goes down into the T-tubules, where it comes very close contact to sarcoplasmic reticulum, and we'll talk about that in the next phase. It'll release the calcium. So just before mentioning that, we also talk about how relaxation happens at this spot. So it's not the next step. The next step is excitation, contraction, but relaxation we're going to talk about at each step that happens, how relaxation occurs. So relaxation happens when we remove that acetylcholine and close this channel. Okay, and two ways that can be. The acetylcholine can diffuse across. Another one is equally important is this enzyme acetylcholine esterase breaks down this acetylcholine. Hence, this channel can now close. Okay. By the way, if you've heard of sarin gas, that is used. That's been used by. It was used by Saddam Hussein. What it does, it inhibits acetylcholine esterase, which means acetylcholine that was released will not be broken down. It will remain bound. So this person is going to the muscles are going to be remaining in a contracted state. So let's talk about the next step. Next step is called excitation contraction coupling. So AP, short for action potential, that moves down the sarcolemma and into the T-tubules. It goes into the T-tubules. It causes basically calcium to be released from the sarcoplasmic reticulum into the sarcoplasm. So this is going to be key. Okay, so action potential comes down, voltage gated protein molecules change shape, which causes calcium to be released. Calcium goes and binds to troponin. Okay, binds to troponin. Troponin holds tropomyosin, and tropomyosin is blocking those active sites. When calcium binds to troponin, it changes shape. It moves tropomyosin away. Now you can see those indentations. Those are the active sites of actin. The myosin head can bind to those active sites. Okay, and then this starts off excitation because action potential came in, it triggered calcium to be released, and we start off the contraction process. Again, we're taking a side step. How do we terminate contraction? Again, this is not the next step of the contraction process. So first is activation, next is excitation contraction coupling, and the third and last would be the cross bridge formation. So how do we relax it at this phase is we have to actively pump the calcium back. Okay, so we need ATP for this. If there is no ATP, which is what happens in rigor mortis, calcium is not pumped back. It remains in the sarcoplasm, which means the tropomyosin is not blocking troponin. Tropomyosin is not blocking the myosin heads from binding to actin, so they remain active. They can bind to. Another reason why rigor mortis happens, a twofer. We also need, which we'll talk about next, is we need ATP to be present for myosin heads to unbind from actin too. Okay. So let's talk about cross-bridge cycling. So I'm going to start with this position here when ATP binds to myosin. When it does bind, we're going to first, that's also detachment, but if we start at where we had We start at excitation, contraction, coupling, so we just expose the active site of actin. So we need ATP to bind to myosin head. ATP is broken down to ADP and phosphate. That's where the energy is stored between this bond. So it transfers the energy to the myosin head. It's called it becomes activated, or even the position is like a rooster. The head kind of tilts up, and it can bind to the active site next. Okay, which is cross bridge formation, and this happens when we release the phosphate, when we release the ATP from it. The next is called the power stroke, where we are going to, this myosin is basically going to go back to its non-activated position because it's lost its ADP and phosphate. It's going to pull, so you're going to imagine that M line is somewhere towards the left here. So it's going to pull the actin, the thin filament, towards the M line. Okay, so Z disc will be somewhere here. M line, of course, this is not to scale, but it's further towards the left, right? So Z disc is further towards the right, and it keeps doing that. And for this to be detached, we need ATP. If there is no ATP, this will remain attached, rigor mortis, right? So when we die, when anyone passes away, they go into a state of contraction, a catatonic state. Okay. So contraction cycling repeats as long as ATP is available and that calcium remains high. So if we pump calcium back, then relaxation will occur. Contraction coupling will not take place. Please note, so ATP is required to disengage, to remove the myosin from actin. It also is used up to energize the myosin head. We also need another molecule of ATP to pump, or molecules of ATP to pump the calcium back into the sarcoplasmic reticulum. So, termination of contraction, we talked about removal of ACH, acetylcholine, in the neuromuscular junction by acetylcholine esterase. Then we talk about at the excitation contraction phase, we want to actively transport the calcium back into sarcoplasmic reticulum. And in the next phase, we need the ATP to break the cross bridge of the myosin head. Okay, so once these happen, then actin and myosin detach. The actin filament slides back away from the M line towards its relaxed position. So I hope that was helpful. Have a great day, and I'll catch you in another video.